Hello everyone. So today we are going to start our biochemistry lecture from the USMLE Step 1 book. So when we start this USMLE biochemistry, we see the first topic is chromatin structure. So first of all we will deal with the chromatin structure. So let's see what it says here. DNA exists in the condensed chromatin form to fit into the nucleus. DNA loops twice around the histone octamer to form a nucleosome. H1 binds to the nucleosome and to the linker DNA, thereby stabilizing the chromatin fiber. So when we study this, we find that it is not giving a clear picture of what is a chromatin structure. It starts with DNA exists in condensed chromatin form. So we must elaborate it. So let's see what are the things we are finding here. First of all, they say DNA, then it says chromatin. So are these two are a different thing? Like it says DNA exists in condensed chromatin form. Are these two are a different thing or a same thing? So let's discuss. Let me make the chromatin structure here first. So just look at this. This is how a chromatin usually is. This is a normal chromatin structure. Okay. So for to understand its structure completely, we have to zoom it here. Let's take one side here and let's zoom it. So it is like this. So what it actually is, let's make the other layer here. We have made this is usually like a double layer. Okay, I will explain what this double layer is, but it is a double layer. Just remember it for now. So we have made this a DNA double layer. Okay, it starts with DNA. DNA exists in condensed chromatin form to fit into the nucleus. So basically what is happening, DNA is coiling like and is in condensed form. Why? So that it can fit into the nucleus. So what are these other things here? It loops twice around a histone octamer. So we have to make a histone octamer here. Where is the histone octamer? It is usually here. Okay, this orange colored is a histone octamer. Okay, what is this? Histone. What is this? DNA. Okay, so what is happening here? This is a histone octamer. DNA loops twice around this histone octamer. What is this histone? It is a protein. Okay, so DNA has looped twice around this histone octamer to form what? What is this in total now? This is a nucleosome. Nucleosome. So what is happening here? DNA is coiling or looping twice around this histone octamer to form a nucleosome. This whole now is a nucleosome. Okay, so is this a chromatin now? No, it is not. We have to see there is one more thing. H1 binds to the nucleosome and to linker DNA, thereby stabilizing the chromatin fiber. There is one another thing here. This is H1. It is around here. Okay, so what is this H1 actually? H1, H1. Okay, so H1, just see this is a nucleosome. There is a histone and DNA is looping twice around it. Now this DNA has to combine with the other DNA, other nucleosome. this nucleosome has to attach to the other nucleosome, so something has to there has to be something which has to link with it. So this H1 is a linker protein which binds from one DNA to the other DNA. It starts from one, it attaches to the nucleosome, and it's on the other side the other DNA attaches, and thus it is forming, it is attaching these nucleosome, and there is a string of nucleosome is formed. Okay, so now what we understand, histone DNA loops twice around it to form a nucleosome, and H1 binds to the nucleosome and to the linker DNA. Okay, so now can we say this only this structure is a chromatin? No, we cannot say that this whole structure has to be chromatin, because why? Chromatin includes 10 to 15 million nucleosome. Okay, a chromatin includes 10 to 15 million nucleosome. I have made just 4 nucleosome here, but actually a chromatin includes 10 to 15 million. Okay, so now this whole structure is a chromatin. Okay, so now everyone must understand what is a chromatin. Histone protein DNA loops twice around it, and it is attached to the other DNA by this H1 protein, and now this whole forms like this. Now let's zoom in. It is just like this. Okay, now this chromatin can easily fit into the nucleus. DNA is arranged in a condensed form so that it can fit into the nucleus. Now this whole structure is called chromatin in medical language. Medical gives names, so it gave this name to it. Okay, so now this is chromatin. Now there is one another thing. Now look, this is a chromatin, and this is another structure that you guys must be familiar with, and you must be getting confused as well. This is chromatin, and everyone knows this is chromosome. Okay, this is chromosome. So now what is the difference between chromatin and a chromosome? Are these two same thing, or are they a different thing? Okay, so let's discuss where is this chromatin. This is a nucleus, chromatin is inside this. Okay, so where is chromosome? Chromosome is also inside a nucleus. Okay, in condensed form. Now what is the difference? Are they two different things? The answer is no, they are not different. Chromosome is when the cell is not dividing, then the DNA is in chromatin form. Okay, when cell is not dividing. Okay, and when cell is dividing, when the cell is dividing, it is in chromosome form. When the cell is not dividing, then it is in chromatin form. So now what is the difference between these two forms? Why they are in two different forms when the cell is dividing? Because chromatin is not that tightly packed, and chromosome is very tightly packed and very condensed, so that it forms this structure. When it forms this structure, then it is easy to divide the cell. Half of this part can easily go out, so it is very easy to divide the cell. In chromatin it is not easy to divide the cell. It goes into a condensed, hugely condensed and constricted form, so that it can divide easily into two parts. So we are not going into the detail of chromosome structure. We are just explaining the difference, because most of the students gets confused between chromatin and chromosome. So remember, they both are just the same thing, nothing is different. So they both are same thing, nothing is different. Just one thing, when the cell is dividing, They just change their structure into a a highly condensed form so that it can divide easily. And chromatin is just a little bit in loose form than the chromosome. It is just only that tight so that it can fit into the nucleus. Okay, so if you want a detailed lecture on the structure of chromosome or DNA, then you can comment below so that I can make one. So now we understood everything. DNA exists in condensed chromatin form to fit into the nucleus, a condensed chromatin form, so that it can fit into the nucleus here. Okay, DNA loops twice around a histone octamer. Okay, DNA is looping twice around a histone octamer to form a nucleosome. H1 binds to nucleosome and to linker DNA, thereby stabilizing the structure. If H1 doesn't bind to these DNA, the structure will not be stabilized. Okay, so what is left here? DNA has negative charge from phosphate group. Histone are large and have positive charge from lysine and arginine group. So what does this mean? Positive and negative can combine, can form a structure, can form a bond. Okay, so it is nothing to discuss in here. Now DNA and histone synthesis occurs during S phase. This is during the mitosis and meiosis. So we will discuss this in detail in a separate lecture. This is not a part of chromatin structure. So now there is just one different thing, that mitochondria have their own DNA which is circular and does not bind histone. So there is just one thing left which can be important, that mitochondria have their own DNA which is circular and does not bind histones. What does this mean? That DNA doesn't bind with histones, so it is circular and it is not in this normal chromatin structure. So if someone asks you that is there any DNA which is not in chromatin structure in your body, then it is mitochondria. Mitochondria doesn't have a DNA in chromatin form. It is in circular form and does not bind histone. So this is just the only important topic here which can also come into the exam, and all we discussed before was just to understand its structure, so we have a clear concept and we doesn't falter about or we just doesn't forget about it. Now this can also come in exams, some this type of question. If something is exam type material here, then this line. Okay, so just remember this line, that there is one DNA which is different and is not in chromatin form, that is the DNA of mitochondria. Okay, so if you didn't understand anything, then you can comment below, and if you want lectures of in detail about any other thing, then that you can also comment below. Okay, okay, thank you so much.